All right, Marco, I can see that.
Yes, sorry. Now I should be working fine. You can see the screen, right? Yeah?
Yeah, we can see the slides. We can hear you speak. All good.
Perfect. Yes, sorry about that, but thank you very much for the invitation and for giving the chance to present a little bit of our work. So yes, I will talk about fish and the possible role that the forebrain has in regulating the stress and emotional responses. And I'll start with basically just a little bit of an introduction of the HPI axis. So similar to the mammalian, but in this case, it has an I instead of an A, and it's because the interrenal tissue is where the cortisol is being released from. But it basically works the same way as in mammals. And just as in mammals, we have a negative feedback loop. It happens at different points, and basically is to regulate the system. So this is like very standard. And we also know that there is, as Tom already introduced, there is definitely some regulation from the forebrain. It's a little bit not completely known exactly some of the methods, how is this happening, particularly on fish. But what I'm going to be talking about today is the role of the serotonergic system. And I'll share with you guys a little bit further down the line what we believe is the major player in regulating this the stress response. But this was introduced, at least in fish, a few years ago by Svante Winberg in Uppsala, where they talk about the fact that in fish is pretty much like as in mammals, where there is this co-regulation of serotonin and cortisol. Basically, both regulate each other. So, of course, we have to talk about the comparative side of things here. Obviously, a fish brain and a human brain are very, very different if you just look at them this way. But of course, once you start looking at different parts, we do see that there are a lot of similar areas, and a lot of them have similar functions, even though they might be placed in different parts of the brain, so to speak. And what I'm focusing on today is mainly these brain areas, mainly what we call the amygdala, the dorsomedial pallium in fish. But as I said, I'm going to be focusing on serotonin. So a little bit of introduction of what I mean when I talk about serotonergic activity. And that is, as you all know, we have vesicles that have the neurotransmitter serotonin in the presynaptic neuron, and they are released and they can be taken in by receptors in postsynaptic neurons. So what we measure is the concentration of serotonin, but that basically means that you're measuring what's in the synaptic cleft, inside the vesicles, basically everywhere. But at the same time, to get a better idea of what is the release and signaling of serotonin, what we do is we also measure the main catabolite, 5-hydroxyindoleacetic acid, which is basically the degradation of serotonin that has been outside and is pumped back into the presynaptic neuron. So what we do is we look at the concentration of the catabolite, we look at the concentration of the neurotransmitter, and we look at the ratio of this. And this gives us an idea of how much has been released. I will also talk a little bit about the 5-HT1A receptor, which is what it is believed or has been proposed, at least in mammals, that has to do with the regulation of stress and cortisol. So to start off with, I will talk a little bit about coping styles. Maybe you are familiar with it. And basically, it's the idea that individuals consistently respond to stress in the same manner, both behaviorally and physiologically. And we have mainly two types of coping styles. The reactive, that is a little bit basically inhibited, not doing much, sit and wait. And we have the proactive that is a little bit more forward, more active. And there's plenty of research that has been done now in fish, where we have found that we have both reactive and proactive coping styles. So we set out a few years ago to select for coping styles by basically looking at how individuals respond to diminishing levels of oxygen. Basically, as the levels decrease, the individuals could move to another tank, and they chose to do that at different points. So in the end, we ended up with the tank where we had the ones that left, the levers, and then we have the stayers. And this is basically sort of the levers would be the proactive, the stayers would be the reactive. Now, behaviorally, this is what they did. We also look at the physiological response by looking at stress cortisol levels. And then usually what we have is less stress levels in proactive than in reactive, and this is what we found here. So then we decided to look at what's going on in the brain, particularly the forebrain. But one of the things that we've noticed throughout the years is that the fish research tends to be a little bit behind the mammalian research in terms of how specific we look at neuronal populations. When you look at a lot of the research that has been done, and we've done ourselves this, often you look at macro areas. So you look at the whole telencephalon, for example. So we wanted to see how much information can you get from looking at the whole telencephalon and compare this to some specific areas. So what we found is that, okay, we have the levers here in green, we have the stayers in blue. And what happens with the serotonergic activity? Yes, it increases basically if they experience an acute stressor. Great. Doesn't appear to be any differences between the levers and the stayers. However, when we microdissected the dorsolateral pallium, which is the hippocampus, or the functionally similar to the hippocampus, then we see all of a sudden that there are actually some differences between the levers and the stayers. And then when we looked at the dorsomedial pallium, the amygdala, then we see that there are some differences, particularly after stress, between the levers and the stayers. But it was even more interesting because in the case of the stayers, they don't seem to react with an increase of serotonergic activity, which is what you are seeing on all the other cases. So then we also looked at the 5-HT1A receptor. And this is what we find if you look at the whole telencephalon. You basically see a downregulation of this receptor in the levers. But then if we look specifically at, in this case, the DM was the only one where we found any significant differences. We see that there's actually an upregulation. So it's very different. If you start looking at specific nuclei, you see a different expression, different results, which is obviously not surprising. When you look at the whole telencephalon, you're looking at a lot of different neural populations. So you're getting a mix and match of many different things. So it's important to look more specific into the brain. And then that's what we did. And we looked at a bunch of different monoamines, serotonin, dopamine, noradrenaline. We look at different genes for serotonin, for stress, the stress axis, and so on. And this is a bit of a complicated figure. And I'm not going to go too much into details here, but one of the things that I want to point out is that, again, we see a lot of interesting things happening here on the DM, the amygdala-like structure, particularly when it comes to serotonergic activity. And this is what I want to go back to. First of all, we're going to go a little bit back in time. We're going to look at something else that we did a few years ago. Is we looked at this so-called depression-like state fish. It's a fish that you find in the aquaculture industry, where a small population of fish act like this. Basically, they are not very reactive to things. They eat little, and they are quite thin, and most of them end up dying. So we wanted to characterize this fish and try to figure out what's going on. So what we did is that we sampled them at basal levels and then after stress, and we looked at the serotonergic activity here first. This is a healthy individual, and you see that this is their normal, their basal response in terms of serotonin. If you stress them acutely, they increase the serotonergic activity. And now we're looking at the brainstem. So I have to go, even though this is about the forebrain, the story starts here in the back. Then when you compare this to the DLS fish, you see that they already show at basal levels signs of chronic stress. So very much elevated serotonergic activity, and they are not able to increase this when they are challenged further, which is bad, because serotonin acts like as a buffer for cortisol. So if you have an increase of both cortisol and serotonin, there's a lot of buffering that happens in the brain to combat the negative effects of too much cortisol over a long time. If you don't have that extra buffer, then you may run into problems. We also looked at the cortisol response, and we basically found the same. So there is here something that I'll go back to in a little bit, which is there is the co-regulation of both serotonin and cortisol. There is a correlation there in which it should be a positive correlation, but I'll come back to this in a sec. So then we decided to see if we could create some DLS fish ourselves by chronically stressing them. Basically by increasing the carbon dioxide in the tank over a few weeks. And then we looked again at the macro areas. We looked at the forebrain, we looked at the hypothalamus, and then we look at the brainstem. And in the brainstem, we see exactly the same pattern as we had found on this other DLS fish. There seems to be some regulation here in these other areas, but nothing that was very remarkable in terms of the forebrain. But as Tom already mentioned, the forebrain does process a lot of the stimuli that comes into the system. So therefore, we wanted to see what's going on there in terms of the expression of different genes. But before we go into that, like I mentioned before, this is the correlation of cortisol and serotonergic activity. As you can see, this is very much a positive correlation, but in this DLS fish, we don't have the correlation anymore. And this I'll come back to in a sec. First, we're going to look at the different expression of genes in the telencephalon of this fish. This is the healthy individuals. This is what we found in differentially expressed genes when you respond to stress. So there's a lot going on, as you would expect. Some of them are downregulated, some of them are upregulated. So what happens with this DLS or CO2 fish? There's not a whole lot of happening there in terms of how they are responding to stress. So this shows a system that is malfunctioning. Then we look at the differences between basal and CO2 fish, and again, not much happening. But then when we look at stress for both groups, then we see a few more things happening here. And then if we look further into those genes, and I'm not going to go too much into detail here, but basically what you see here is the control group at basal and stress levels show a downregulation of a few of these genes, while if you compare between the both stress groups, you have an upregulation of those genes. And what does that mean? Well, if you look at the trends here, you basically find the same thing that we found for serotonin. You have a system that at basal levels at least appears to be a little bit higher, and when it comes to stress, the DLS fish do not respond further, while the control fish respond with increased activity after stress. So again, we're now showing that in the forebrain, the expression of a bunch of different genes that regulate a bunch of different processes that I'm not going to go into details now, but it is being regulated in a similar manner to the serotonergic activity and the cortisol on this DLS fish. So then we wanted to figure out what happens if we try to manipulate the system, particularly with fish that are in the freshwater phase at early stages. So already when they are going to vaccinate fish in the aquaculture industry, if they have any fish that are below 30 centimeters, they discard them. Then they vaccinate, and if they don't reach a certain size, they don't send them to the seawater phase. And that means that these two groups are potential DLS fish. So we decided to work with the smallest ones. What we want to know is, because we know the serotonergic activity is increased in DLS fish, so we wanted to use buspirone, which basically decreases the serotonergic activity. So we used buspirone in the water. We subjected them to a bath for basically an hour. And then we observed the fish, and then we took some samples. And the most interesting thing that we found here was that, again, in the DM, you do see that this system is being downregulated, particularly. Something that I haven't mentioned is that the buspirone actually acts on the 5-HT1A receptor, which I mentioned earlier is what it is believed is the main regulator of the stress response in terms of serotonin and cortisol. So again, this DM keeps popping up in our research. And just quickly, there's a few other things that we have done where, again, the DM has popped up as an interesting area. And it is, for example, this experiment that we did on chronic social stress, and then they were subjected to ketamine, which increases the activity of fish. This was before they were going to meet a new dominant fish. And then we, again, saw that there were differences in the DM. We also see this in early life stress research. Basically, fish were stressed during the first or during early in life, and then we saw how this affected fish later in life. And again, the DM popped up as an area that was highly regulated. And we also saw this in terms of frustration. If fish were expecting a reward and they didn't get it, there were changes particularly in this area of the DM. Now, more recently, we've discovered something interesting. And I first have to go back into the midbrain, even though this is supposed to be about the forebrain. But this is an example of Arctic char in the wild. These fish have some parasites that basically attach to their eyes. And an interesting thing that happens is that these fish are sexually mature, and the ones that have the brightest colors and that are most bold, basically flashing a lot of the surface and stuff, they have the most amount of parasites, which is interesting because these fish get predated by birds, and the bird is the final host for the parasite. So it may be possible here that the parasite is manipulating its host, in this case, the fish, to be eaten by a bird. What we saw was that when we look at fish that have high amounts of parasites and low amounts of parasites, I think it's a little bit hard to see, but basically there is a lot more neuronal activity happening in the posterior tuberculum. We looked at the whole brain, and it was specifically this area that had dramatic differences in terms of the neuronal activity. And we're talking basically all these areas throughout the midbrain. And when we sequenced these areas, we found a few things, but in general, very interesting that the GnRH signal pathway, the gonadotropin releasing hormone pathway, was differentially regulated between these fish. And mainly, there's a few things going on here, but I'll focus on the upregulation of the GnRH gene and the luteinizing hormone gene. And I focus on those because what we believe is happening is that GnRH is being stimulated in this area in the hypothalamus, which in the pituitary leads to an increase of LH. And this could be behind the conspicuous behavior that you see increased in this fish and the increase in risk-taking behavior plus the increase in coloration. Again, is this parasite manipulation or just the fact that fish that are more sexually mature have a lower immune function and therefore they have more parasites? We still don't know, but this is something that we're working on. And again, this area was quite interesting that it popped up this way in this research, because it happened again when we're looking at how sound stress affects salmon. Basically, we looked at different time points before salmon are being subjected to the sound stress. Then the first time they hear this sound stress, after 10 times, after 20 times, after 30 times. As you see, this is the cortisol response is highly increased the first time they hear this sound stress. But what happens in the brain? Well, what we saw, again, we looked at neural activity throughout the whole brain. And then, again, we saw that areas close to this tuberculum, the posterior tuberculum and adjacent areas were differently activated in fish that had experienced and not experienced this sound stress. So again, this is quite interesting. However, we also saw that in terms of the DM, it is also a lot going on in this fish. So then what we are thinking is that there might be a specific connection between these two areas that is being regulated during stress. So what we want to look at further is take some microdissections, look at these two areas, and then try to figure out what sort of mechanism may be at play there. So some concluding remarks. Serotonin regulates the stress response, and it appears that this has to do with the 5-HT1A receptor at different levels of the brain, but definitely that it is involved. The DM appears to be extremely important in stress and emotional responses. Now, I gave you a few examples, but there's actually, we've seen it in other contexts as well, in terms of increased exercise training, in terms of reward expectancy. This area keeps popping up. And the posterior tuberculum appears to be particularly important in terms of the stress response in the midbrain, and there might be a link between this area and the DM that we need to look at further. And with that, I would like to thank you for your attention, and any questions, I will be happy to answer.
Thank you very much, Marco. I'd just like to remind people that if you have questions, please type them in the chat where we will see them. We have plenty of time for questions. In the meantime, Marco, while people are typing, let me ask you a question out of basically ignorance of fish brain anatomy, maybe even of vertebrate comparative anatomy, which I shouldn't admit to in this group. The tuberculum in the midbrain, what is that doing generally, and what is it equivalent to in, say, a mammal or a bird?
So mainly it again, this is what we know so far. It has to do with reproduction. It has been proposed before, but this has been debunked, I believe, at the moment, that it had to do also with reward stimuli. Basically, sort of like in mammals, we have the VTA, the ventral tegmental area, which you don't find in fish, and they believe that this could be part of that area in fish, but it is now found that it doesn't add up basically. And as far as I know, I believe that it is actually the same. It's called the same in mammals, if I'm not mistaken.
That's great, thank you. David has a question, and actually this is another way we can go. If people want to just put their hands up, I can allow them to unmute and ask their questions directly as well.
Thank you, Tim. Yes, Marco, I had a question concerning one of your first slides, coming back to the experiment when you have these two water tanks with the stayers and the levers. I was wondering whether you tested or not the differences, interindividual differences in terms of sensitivity to hypoxia or resistance to hypoxia, because if some fishes are more resistant or less sensitive to hypoxia, perhaps they would stay in the tank and not go in the other one. Yeah,
that's an excellent question and a very good point also. We actually did not test that, but this was done, it's been a few years now, but I believe it was 2 or 3 times to look for consistency. So the fish that left always left. The fish that stayed always stayed. But because this is, I mean, again, we correlated this to their stress response, and the fish that stayed always had higher cortisol levels, which would imply that they are not staying because they're necessarily resilient, but because they're sort of inhibited in their behavior. But of course, it could be the case that they are just more tolerant. I would expect, though, that if they are more tolerant, then they would have lower cortisol levels, for example. Yeah. But again, it's not. We did not go into every specific individual to look at some of the difference. We just looked at the extremes at this during this experiment.
Thank you.
Yeah.
Hey.
Me.
Yes, go ahead.
I have, first of all, that it's a rather comment than question. That posterior tuberculum, why do you consider it midbrain?
Yeah. Sorry, I didn't get your question.
Why do you consider the posterior tuberculum as the midbrain?
Just because it's part of the hypothalamus. And sorry?
It is considered to be the forebrain.
Okay.
It's rather the ventral part of the diencephalon, and there's no, well, in the equivalence in amyloid, it's rather small, and this function of a reward system, I don't think so.
No, that's right. What I mentioned is that it was believed to be that.
Yeah, and
it was debunked a few years ago.
Yeah. So then you mentioned about the posterior tuberculum, and at the same time you talk about GnRH, because the yes. GnRH is not located in the posterior tuberculum. And
there is some evidence now, some relatively new evidence that there are GnRH populations within this area of the brain.
Where exactly?
I can send you the paper if you send me a.
Okay, yeah, I'm curious about knowing that.
Yeah, it's something that we were also quite surprised by when we got the results, and then we started doing some research, and then we found that paper, for example.
But then at least these populations, they are not sending the projection to the hypophysis. I think in fish, the direct projection to the LH and populations are from the preoptic area, the most anterior part of the preoptic area. So yeah. It wouldn't be the same population, the controlling the reproduction.
This is relatively new research that we're doing. So if you say so, because we haven't gone into that yet. We're looking at what could be the possible pathway for this. As I mentioned before, this is something that we believe could be the pathway that I showed, but this is very early stages. So we haven't looked into all of this yet.
Okay, because so far there is no data showing that I think it's rather the projection to the pituitary is quite specific. So
Okay.
I don't think there are some posterior tubercular cell population projecting to the hypophysis.
So, but you're saying that it goes through the preoptic area.
The preoptic area in fish, because the preoptic area in mammals and fish is not exactly the same. But there are population of GnRH projecting directly to the hypophysis in the most anterior part of the preoptic area.
Okay, but then we'll look at that as well, because as I mentioned, this newer this research that has been done in terms of the GnRH population within this area are something that we are heavily basing our research on. So we need to look at some trace studies and try to figure out what would the connection be. But thanks, it's good to have this feedback.
Okay, well, thank you.
Yes.
All right, thank you very much. There's one question in the chat and then we should move on. Amalia is asking, did you look at any effects in the subpallium?
In terms of which?
I guess in terms of the stress responses would be my guess, Amalia.
Well, I mean, in terms of the stress response, yes, we mainly look at the ventral part of the ventral telencephalon, which is believed to be functionally equivalent to the lateral septum. There are some interesting, there is some interesting regulation happening there as well, but it hasn't been as consistent as, for example, the DM in terms of how active it is or not. Of course, it may be more specific to different responses. Yes, it was the stress response. And in terms of, for example, reward expectancy, we definitely see a lot more activity going on there, particularly those areas where you have dopaminergic neurons.
Great. Okay, well, I think we should leave it here. Thank you very much, Marco, for your presentation and for everybody for the questions, and Marco